Welcome, everybody. My name is Jelani, and welcome to the 47th episode of the DeSci Rising series put on by DeSci World, where our goal here is always to, among many other things, provide an opportunity for many of the wonderful projects on our growing DeSci World dashboard to just kind of formally introduce themselves, update their progress, highlight their achievements, and just generally engage with both the DeSci World and the wider DeSci community. Now, the idea behind all this is that at DeSci World, we very much believe that more than the tech, more than the funding, it is the community leveraging these tools that will usher in many of the changes that we're all looking for DeSci to bring to the various legacy STEM industries. Now, in today's episode, we have a really interesting conversation, which is going to span the divide of what I just talked about. We're going to be looking at tech and applications, but also the human components that are vital for pushing science forward. And so, with my pleasure, I have a pleasure of introducing Shar Qureshi, who is the founder of Qure, which is a platform that is pioneering transformation in the life sciences, bringing about an immersive, collaborative metaverse where scientists can enhance their skills and fuel groundbreaking scientific discovery. I think to their own platforms mentioned, this is all in application. I mean, its applications are very wide, but specifically, this was going to help hopefully combat the replication crisis, which is an epidemic that I think is really underrated in the concept of the world and severely needs addressing. So, with all of that Gabrajaw being said, welcome, Shar. Thank you for taking the time to be with us today and to help us learn more about what you were doing and what the great work is being done at Qure.
Thank you, Jelani. Really happy to be here. Can you hear me all right?
Yes, I can hear you great.
Great. Yeah, great turnout. Thanks so much for the opportunity.
Yeah, I think, and just to the rest of the crowd, we want to make this really engaging and pluralistic affair. So if you have questions, you have comments, please don't wait till the end. Put up your hand, I'll bring you up on stage and you can join the conversation. I think Shar has a wealth of information and knowledge on his own end. And I think this is going to be a fantastic conversation. So the way I think I like to, we all should start this off is just by a little bit of an introduction to you, Shar, and not necessarily from the scope of Qure, but I'd love to hear your origin story of where you came from before you got into this space.
Yeah. So, I started my career in science. I was a biology major. Was minoring in Chinese at the time in Ohio, and that's where I really fell in love with life science research. It started out in retinal regenerative research, which was really, that's what kind of made me fall in love with molecular biology and understanding these pathways that may seem so chaotic, but we're able to simplify them down into a pathway. Whereas within the cytosol, it's mostly a chaos and soup, but these molecules never find a 404 error, and we're able to function amazingly. So I fell in love with the laws of nature, and the study of it. So I continued on that path with a graduate degree in nanotoxicology. Worked with the toxicology studies for the Air Force, the United States, for a number of years before I moved to Boston and got into the world of biotech, where I was a scientist and then a field scientist where I trained other scientists all around the world. And that's really where I got my first exposure to some of the inefficiencies in science, training other scientists, standardizing certain protocols. This still, in 2023 is an obstacle. And the standardization and ability to reproduce science is kind of the foundation of the scientific method. And so that's really where my first passion for reproducibility came, and seeing that working with some of the most brilliant scientists in the world, at the Air Force and then in Boston, and training other brilliant scientists, what I found was that there's a huge disparity between their ability to promote science and life-saving and life-changing technologies and research, and the disparity between their education level and perhaps their compensation. It really didn't sit right with me. These people are at the forefront of innovation in almost every sector, but they certainly are given the back seat in terms of monetary compensation, and are told to kind of be complacent with this publish or perish culture of seeking clout um, by publishing in prestigious journals, and that's amazing that it still happens, right? And that quality research still is produced, even with this disparity. So, with that background, the executive team at that biotechnology company thought I'd be better fit in sales than in field science. So they moved me into a new sales territory. That was my first kind of role on the dark side, if you will, away from the bench. Um, and through that experience and many other kind of more business related experiences over the last 5 years, I founded Qure as I my passion for the lab never left me, and my passion for innovating science and wanting to reward scientists for their contributions to humanity was really magnified, and that's what led me to founding Qure.
I think that's a fantastic origin story. I think many of us who have found ourselves in this space immediately face some of the challenges directly, whether it be the lack of standards, which is such an interesting thing in the realm of science, or
Mhm.
misaligned incentives from the funding agencies or publishing and the issues that exist there. These are all firsthand issues that I think many of us have dealt with. And so, to your point of the lack of standards, because I think replication crisis is really underpinned by largely this lack of standards. There are a few other things like the monetary policies that are at play, the publish or perish culture, but I think standards across labs, between labs, across databases is one of the biggest issues that exist here. And so why do you think we have this issue with standards? What do you think is fueling this fact that there's lack of interoperability, lack of common language used among scientists and different science groups?
Yeah, that's a that's a big question. Right? There's a lot of facets here that play into reproducibility, into how we've gotten ourselves here. Much of them are systemic. Much of them are maybe regulatory based, um, and you know, standards at a foundational level, I think, um, is accounting for human variability and human error. I think variability is a better way to go about it. But that variability can vary in degrees, right? It can be very different to slightly different. Um, but Leonard Friedman, who's now at the NIH, he wrote an excellent article in 2015 called "The Economics of the Reproducibility Crisis," and I heavily cited his work in Cure's first blog post, which is here on Cure. It's on our Twitter page. Uh, that kind of outlines the four key drivers of reproducibility, which include protocols, the bioreagents used, um, and equipment standards, and human variability. So these are four major drivers of the reproducibility crisis, um, and I think it really comes from where we are today. Looking at a macro view, this publish or perish culture, people become very complacent to think that this is the way things are done. And I'm a pragmatic contrarian to that idea. Um, in what I said earlier, these guys are changing the world. Their education is leaps and bounds over many other industries. However, their incentives and compensation does not align with the impact that they have on humanity. And so I think DeSci is causing scientists to open their eyes to this disparity, um, and that reproducibility really does have something to do with it. Because if we're functioning under the boundaries of publish as much as you can, publish in a higher tier journal, really focus on the high impact stuff, we may lose a lot of kind of research points by focusing in within the boundaries of the publish perish culture today. And so, the incentives are just misaligned. And I think that that really impacts reproducibility and from a macro view.
I like how you put that. The target really is publish high impact, but the kind of misnomer or the paradox that exists there, and it's not pragmatic impact. It's arbitrary impact, as in which journal you're in and the impact factor of said journal, but not really how it affects individuals at the end of the pipeline, at the end of the research, at the end of the therapies. And I think to your point, what we start to see now, or what we're hoping, or what people are starting to build, is these KPIs that allow us to actually determine or look at impact from the target populace, and then retroactively determine which science is truly impactful. And I think as that starts to develop itself and we start to be able to look at that in real time, as more databases, as more data entry, as more data structure, especially interoperable, starts to come online, we can actually start to re-look at and re-envision and retarget impact based off of true, realistic impact and not this arbitrary metric. Now, to your point, you mentioned there are a few pillars of aspects, or there are a few pillars themselves that help prop up this replication crisis. So let's delve a little bit into Qure. What is Qure, and which of these pillars is it looking to tackle?
Yeah, so Qure is a life science metaverse. So we're creating a home for scientists through extended reality. Currently, our focus is on virtual reality, but we have every intent to move into what Apple showed as their big shiny future into mixed reality, and the level of standardization that is able to be met today. We're in 2023. We really got to think about that. The current way of doing things is getting us the current results that we have, right? And replication is a major crisis. I mean, this is the scientific literature, the ledger of science is being kind of moved towards a bit of a carousel. So Qure aims to solve much of replication by focusing on two of the four drivers of reproducibility, which is the laboratory protocols themselves, as well as biological reagents. Um, this is through virtual reality. So the goal of our platform is to create a protocol generation platform right now. That's our V1. So a scientist is able to conduct any uh, experiment inside of virtual reality, record how their hands move when they're executing this experiment. So imagine it as a master class, right? If you're looking to learn how to make Gordon Ramsay's famous scrambled eggs, right? There's one thing to watch the video and reproduce his world class scrambled eggs from a video. The chances of you reproducing Gordon Ramsay's scrambled eggs just from a video maybe are a little low, right? And the scrambled eggs example is quite weak. But imagine if you were to say it's like a beef stroganoff dish, right? Much more involved, a lot of steps in the recipe, a lot of ingredients in the recipe, right? You can relate that to a laboratory protocol. Many of these laboratory protocols are 14 steps and longer. They take 2+ days at some times to execute, and the reagents used within them are from different vendors in different parts of the world. So standardization already we're fighting an uphill battle. So where Qure comes in is our ability to, at a high fidelity, record uh, exactly how the original scientist executed an experiment, uh, and allows others to learn exactly how they execute that experiment and conduct it in real life. This is all based on muscle memory, right? The more, there's a psychological theorem called the cone of learning by Edgar Dale, states that after 2 weeks, you remember 10% of what you read, 20% of what you see, but you remember 90% of what you do or simulate. And that's all based on muscle memory. So virtual reality's innate ability to facilitate skill acquisition and technology and talent transfer is quite innate. And so we're leveraging that ability of VR to facilitate a high recollection of steps through muscle memory. Um, is the foundation, the scientific foundation of our technology. And we take that a step further by enabling kind of a formal reporting of that protocol, where you state exactly what your reagents are, from which vendor, just as you would from a method section. And our web app actually has, we've integrated ChatGPT's AI into that, which now allows us to, you're able to copy paste a method section from any paper, place it inside of our web app, and it will give you all the steps of the protocol and all the reagents you need for that protocol. And you can edit it and fine tune it from there. So that is the basis of our technology, is a protocol generation platform for scientists, where they're able to share these protocols either internally for their own labs, for their own new hires, or experienced scientists who are learning a new technique, um, or to be able to share internationally your protocol for peer review or even decentralized peer review.
I think, I mean, this is fantastic. So, I mean, you and I have spoken a few times. I think the first time was quite a while back, and I was immediately impressed by it because this is such a non-trivial thing. I think external to people who work in science, scientists themselves and technicians, people might think, well, how necessary is it really to have this muscle memory that when you have a recipe? But for anybody who's actually done an actual procedure, and tried to follow one of these standard protocols, you quickly realize that oftentimes you have to amend things, small things, big things. And when you're generating your own protocol, like oftentimes, give or take a year, that original protocol sheet has like different writing on it, different penmanship, didn't fit it, pen entries. As we start to amend these things, like, oh no, this didn't quite work. We need to adjust this volume and all these kind of things. It really becomes a very personalized way to do science. And each scientist does science and follows the recipe slightly differently, which introduces these concepts of, variability within a given recipe. For those, again, who are not scientists, you can think about it as your grandmother's way of cooking. I don't know about you, but when I ask my grandmother, hey, what's this recipe? Oftentimes she'll point at this book that has the recipe, but when I follow it and I watch her do it, there's deviations. She puts in a little bit of extra paprika or thyme or what
Mm-hmm.
have you. And there's a process to this that is unique to herself. And that
Yeah.
exists in the scientific realm. So the idea of being able to virtually experience an individual going through either whether it be the architect of the original protocol or somebody who is just training you that has had opportunity to use that protocol. I think that becomes really, really important because those small nuances that are not captured in the writing of the two-dimensional writing on the paper, is what can make all the difference in a data outcome from a particular experiment.
Yeah, precisely. And it's that variability, because science is so nuanced, just like cooking, right? Like you said, it's an extra pinch of paprika, right? When you're executing an experiment, or you're culturing primary cells, or you're conducting advanced protocols such as organ on a chip protocols, which take 2 weeks just to set up, and then the experiment itself runs for another 2 to 3 weeks. These are highly involved. I mean, imagine doing the exact same thing as someone on the other part of the world, in an experiment that spans 5 weeks, right? That's opening the door for variability and obvious reproducibility issues. So having this high fidelity way to experience how another scientist conducts an experiment, that should remove many of those tactic learning, those tactic knowledges that are lost when you are transferring a process or a protocol from the original poster or the original scientist who created it, who most likely has a very nuanced understanding of the entire process, to someone who's just seeing it for the first time. You know, transferring that through paper or video or through a picture uh, is not effective. That's what we've been doing for years, for decades. But now we have new technology that enables us to to remove much of that variability, to be on the same page about these nuanced steps. If anybody here has ever uh, you've cultured primary cells, it's very nuanced. You have to be very gentle. It takes 5 minutes, and if you just read about culturing primary cells, you most likely will fail. But if you are shown by an actual scientist how to do that, you will always remember it, because you understand the angle of the pipette that you have to hold it at, how far, how gentle, how much, right? All of that is there. So now we see the ability to remove a side by side scientist training by creating a no risk, no cost environment for scientists to inherit new skills.
I mean, that's a whole other piece, right? If you're talking about the economics of a lab, a lot of time goes into training individuals. And as somebody who's trained a lot of scientists, and particularly in managing primary cells, like a lot of reagents are lost. There's a cost that comes to this. And so being able to defray those costs, as you said, by bringing this to a digital realm where the only time is electricity, really, and the only cost is the time that it takes to put these things together, it saves a lot. And again, that is a non-trivial thing, especially if you think of early career scientists that don't have big budgets, don't have access to large grant funding. That little bit can really make a difference.
Yeah, and a second piece to that, and the value of this platform for scientists, team scientists, for labs, for academic departments. Academia sees every year, twice a year, every semester, undergraduate researchers come into new labs. A lab is only able to support as many new researchers as they have postdocs, PhD students, master students, senior scientists to train and manage these undergraduate researchers. So now with this platform, these senior scientists, these postdocs, are able to create a simulation of the experiment that is their bread and butter of their lab, and hand that to not just maybe 3, 4, 5 new undergraduate researchers, but they can do 10 now, because now we're enabling scale. We're not just doing standardization, but we're also enabling scale for training. So my goal is that once we enter academic spaces, and we're already betaing in 2 sites right now, how can you bring in uh, much more undergraduates to basically prop up as labor and experience to execute research, and my goal is to accelerate innovation, right? This should be an accelerant to the amount of research that is output by a lab. And what I would love to see is, scientists now have trouble keeping up with the amount of endpoints and research results that they get. That's kind of my personal goal.
I think the modalities here are not endless, but are quite broad. And I think it speaks to the fact that the data space, the hyperspace of data acquisition grows exponentially as you move up dimensions. So first dimension to second dimension to third dimension now with the frame of metaverse, there's a lot of data capture and data transference that can happen there at a scale that was never seen before. So super excited about that. So let's talk about the actual platform, right? What are some of the components of Qure? How do I, as a scientist who has a methodology that I want to pass on or to use to develop a training simulation, how do I go about doing this? What's the process?
Yeah, so you would get a Qure license from your lab, either personally or through your lab. You would have a virtual reality headset available to you, and it's remote, right? You can take it anywhere. It doesn't have to be plugged in anywhere. So you can be at the lab, in your office, or even on your couch. Um, just yesterday, I was inside of our most recent build, and I'm playing with $200,000 worth of equipment. Um, just casual training on it, creating protocols. So yeah, the process starts. A scientist comes in. They start on our web app. Our web app has a place where you create your protocol. You give it a description, give it a title, and you list out the steps of the protocol. And our AI feature that we just integrated this month enables you to copy paste or even drag and drop a PDF and identify which experiments inside of that manuscript you're looking for. So it'll parse through the method section, give you some options of these are the experiments that were conducted in this paper. Which one do you want to create a simulation of? And then it'll auto-generate all of the steps and all of the reagents, and you can fine-tune it from there. So once you fine-tune it and you publish it into VR, that's when you don the headset and you then go through. There's a UI right in front of you. It's a heads-up display of step number one, and you, as the OP, as the scientist creating the simulation, creating this protocol, will now start recording your hands. So the first step will be, transfer 50 milliliters of this solution into this flask. You'll press start record, and you'll transfer the solution into the flask, and then press stop record. And now, conversely, when a trainee comes, or another scientist on the other side of the world who wants to peer review the experiment you just conducted, that you create a simulation for, they will come in and they will press start. The first step, they will see 50 milliliters of the solution to the flask, and they will see how your hands moved, right? And they'll see how your hands moved for every single step. You go through that simulation, and now all the research suggests that your ability to reproduce this experiment exactly how you were just trained on it increases significantly, almost 230%. And this was shown in surgeons, right, which is a much higher risk, much more involved kind of protocol and process. And so we expect to see even more significant performance increases in scientists.
What are some of the so we talk about pipetting and all that kind of stuff. What are some of the scientific modalities that are present right now that individuals can use? So the bench work is very poignant, but there's a variety of different kind of bench work applications that exist. Is it also functional for other sciences that are not life sciences necessarily? Like what modalities can we use currently?
Yes, currently it's primarily focused around molecular biology research, right? Any type of life sciences. You know, we were talking a little earlier, Jelani. These protocols, they are more or less standardized if you consider an ELISA or a Western blot, right? Those are not our beachhead. In fact, the biotech company that I was at in Boston called Emulate, it's an organ on a chip company. Their protocols, as I mentioned, are 5 weeks, right? 2 weeks to set up the chips, which is a quad or a pentaculture of different primary cells, which organize into a 3D tissue inside of this organ chip to give high fidelity human in vivo relevance to this advanced in vitro model. These protocols are not standardized. There's obviously a basic protocol, but this is a platform technology, right? It's not like an ELISA. An ELISA is quite straightforward. There are different configurations of an ELISA, and those are, more or less recorded. However, advanced biotechnologies is really where we want to focus, and that's where our early vendor partnerships are. So Emulate is a vendor partner of ours, and we're creating their technology inside of virtual reality, as the protocols are so advanced and so involved. And so by starting there, obviously we have ELISAs as well. That's all functional. You can conduct an ELISA, you can change, you can fine tune an ELISA to exactly how you want it, incorporate different reagents. But these advanced biotechnologies, the biggest problem here is that these advanced biotechnologies lack a equally as advanced training technology. And this is a platform biotechnology, as I mentioned. Platform biotechnologies enable hundreds of applications, like Illumina's NGS, right? Everything has its own NGS library prep, right? They're unique, and scientists can create their own library preps. Similarly, in organ on a chip field, which is burgeoning right now, by the way, any scientist can create a new model, a heart chip, a lung chip, a liver chip, a brain chip, a kidney chip, and these chips have different configurations. For example, the liver chip, you can have 3 cell types, 4 cell types, 5 cell types, all the way up to all 7 hepatic relevant primary cell types within. And so it can become quite complicated. So this protocol generation right now is focused on our partner Emulate. So scientists are able to create their own protocols and share them with others. This, magnifies the ability of emulates technology and emulates community to collaborate, which is, I mean, arguably the one of the most key foundational parts of science, collaboration, sharing these protocols. For example, one of Emulate's users has developed a brain on a chip. Okay, this brain on a chip is unprecedented. It's being published into Nature in the next few months. He created it himself though. The Emulate themselves did not create it, and everybody wants to know, hey, Tyler is his name. Hey Tyler, how did you do that? Right? Am I supposed to just recreate the wheel continuously to try to reach what Tyler accomplished? Use as many resources as possible to get to that pace? No. Tyler has the option to share this protocol internationally with anyone on Emulate's platform. And now they're able to accelerate innovation, accelerate research, because now they have this collaboration potential unlocked that is enabled by Qure's platform.
Yeah, the scalability and efficiency transfer is amazing. And I think also internal, as somebody who has developed stem cell platforms and push differentiation, it is a multi-day process. It takes a lot of time. And I think one thing that we haven't talked about is the internal referencing that this allows. If, given the cost, the permissibility of the cost, if I can run, you know, if I can create a virtual rendition of each time I do my processes, then I can also potentially backtrack and see based off of the different results, well, were there differences in the actual running of this thing? Can I self-check my own self? And I think that would be an interesting feature, if not already thought about moving into the future, again, as costs permit. But to allow individuals to kind of self-train and to self-reinforce the skill sets that they use on the daily.
Absolutely. These are perishable skills, right? I mean, I've been out of the lab for about 5 to 6 years now, almost six, right? I feel very confident in my ability to get back in the lab, but having the opportunity to not take or source someone else's time to train, and be able to reinforce it within myself based on my own findings and on my own research to keep myself reproducible, to keep myself consistent. This is a huge advantage. Um, and we haven't even gone into the peer review functionality of this. That's really where I think our platform has makes the biggest impact on reproducibility. And sorry to kind of shift gears there, Jelani, but I feel like that's really important to know that okay, you conduct an experiment. Let's just say it's a cell viability experiment, right? You have your cell viability experiment, but leading up to that cell viability experiment, there was prep, there was treatment, and then the readout, right? All of this needs to kind of align to get an actual result. So being able to create your protocol, um, and as well as the research results associated with it, and put that up on a smart contract that anybody, anywhere is able to access and bid on and get access to your simulation, as well as the endpoint that you're measuring. They're not able to see the results you got because they have to do it themselves, right? This was the foundation of peer review as laid out by Ibn al-Haytham, who was a golden age scholar in Baghdad, and he outlined much of the scientific method as we know it today. They're not supposed to know the result. They're only supposed to know how you conducted it and what your observation at the end was. So that is exactly how our smart contract is laid out. They will know exactly how you did the research that you conducted. They will know what endpoint you were looking for, and then they will submit their own result. And depending on how close that result is to the original posters, they are incentivized in some way, or receive credentials that, hey, this person not only publishes in Nature, which is a very arbitrary readout of a scientist's caliber, but also, hey, this scientist conducts good science. This scientist conducts reproducible research.
So you touch upon a lot of really interesting things, and by all means, I enjoy the pivot because I was going down that direction myself. I just want to put out another disclaimer. There's a few people who have joined us. This is an open discussion, so if you do want to comment, if you want to join the discussion, please feel free to come up. Just put in a request. But let me dissect that briefly. So let's talk about the interoperability piece, or I should say the open science kind of piece. Is there, so I create a virtual walkthrough of my process. I can, I guess, depending on the stipulation in the licensing agreement that exists, whether it's a private or an open industry or an open institute, I can assumedly put that out there for the world to see. As a user who is interested to either review or to acquire skills, like what is my optionality from that end? Is anybody have access to using the platform, or what are some of the characteristics there?
Yeah, and that's an interesting question, right? Because privacy and data privacy, much of these are kind of set by the overarching institution, whether it's an academic research lab, whether it's a pharma company. You really cannot own your own work, right? You're an employee of that institution. So data privacy, but especially in science, you know, it's interesting because currently a scientist publishes, and what do they get? It's their name, right? They publish their name et al, right? Clark et al, Qureshi et al. This kind of naming is really important. So data privacy, having privacy for the scientist who conducted the research or who created this protocol, uh, that that is a big question, right? How do we balance being kind of KYC'd and having an identity that you're able to hold on to, and others not being able to impersonate you, and say, "Oh yeah, this is the research I did," because your name is all you've got in science. Once your name is tarnished, once you show that you made faulty claims or faulty research results, your reliability is kind of under question. So that's something that we're working through, understanding how to balance the ability of scientists to not only keep it public or private, these protocols, but also to monetize these protocols, right? Tyler, for example, the one with the brain chip protocol that everybody wants. Why should Tyler give his protocol for free to those who are asking for it? For innovation, for basic science, that's awesome. That's amazing, right? He should do that. But he also needs to hold on to his competitive edge. He has the brain chip that everybody wants. So he should have the ability to have a competitive advantage, put up a paywall for maybe a year. Those who are really interested can have access. But and this isn't new, right? Scientists have been able to reach out, say, "Hey, I'll pay you to come to your lab and learn this technique," right? They will do that. So now we're kind of removing the travel from it. We're making it totally remote. You're able to learn this technique remotely. So having the ability to be able to protect your own name is really important, from a privacy perspective. But I feel like I've lost track of the actual question there, Jelani.
No, it's okay. I think you touched upon a lot of interesting things that opened up for more questioning. I guess maybe to tie it back at a certain level, is Cure currently focused on more B2B or B2C models?
It is currently focused on B2B, right?
Okay.
We are totally institutionally related science, right? It is institutional game. But what I love seeing right now in DeSci is the formation of these DAOs. These DAOs are exactly where I expected science to move towards, because I know I wasn't the only one who had my doubts about the current publication culture, the way that science is being done, where monetization is occurring, why is monetization not occurring with the scientists. So these DAOs now allow for scientists to get out of the umbrella of an entity, an academic research institution, a pharma institution, and create their own kind of research-based institution, allowing them to crowdfund. These are the people that they're creating teams. Science is a team sport, so that's why B2B makes the most amount of sense. But as science and these DAOs mature, I imagine we're going to find many scientists who have a brilliant idea, maybe are a solopreneur, a solo scientist, two scientists, three scientists. They're able to crowdfund, they're able to fundraise, and then they're able to conduct the science and have an endpoint that maybe they wouldn't be able to get backing from a traditional funding perspective. So B2B, it makes the most sense as science is a team sport, but as science matures, B2C is certainly something that we want to open up to the world and enable anyone to become a scientist, uh, and learn these protocols, rent lab space, you know, conduct research that they're passionate about, whether it's on rare diseases, whether it's on cancer, anything like that.
Yeah, I think that makes a lot of sense. And to your point of opening it up, like going back to the idea of cost efficiencies, I can just imagine where before each experiment is done, you run a simulation. You simulate the process over and over. You basically have your training day before actually getting to it. And I think, again, that saves so much on time. It helps, as you said, up to 90% of efficiency in terms of acquisition of skills. It opens up the land barriers that exist, the geo barriers that exist, where if I'm located in Canada and this person has developed this in India, a leading researcher, I can potentially have access to that without having to incur the fees of traveling that distance. Um, so I think it's fantastic. I want to continue a little bit on the peer reviewing side. So as you mentioned, the peer reviewing process is lacks a level of efficiency in the current realm of science. And as we start to generate more and more complex ways of doing science, that starts to lag even further behind. But I think to your other point of a lot of the innovation coming out of the DeSci space in terms of novel publishing units, ways that we can capture more and different modalities of data and information within a publication, I think that opens us back up. And so I can envision the day where, you have things like nodes, for an example, or these novel units where you can incorporate this training data, this simulation data in the actual publication itself. And so the peer reviewers, which hopefully will be the totality of individuals that are interested in that particular topic, can not only see your recipe, they can see you perform that recipe. And it even might be an amalgamation of multiple runs of that recipe, right? We have the same, N of three standard of you have to show it's an amalgamation of simulations. And so I'd love to kind of hear your opinion on that.
Yeah, the our platform really harnesses the scientific spirit. Okay, the ability of a scientist. Publications are there for a reason, right? They're supposed to be a packet of information, and also explain why the experiment was done, how it was done, and what they found. Right? By adding the metadata of a simulation of, hey, this is exactly how it was done. Here's a high fidelity view of how this experiment was conducted. That holds so much value for post publication peer scrutiny, right? Which happens regardless. It happens on Twitter all the time. But this kind of puts the onus on the peer reviewer or on the scrutinizer to say, okay, this is what you found and this is what you did. Well, let me do it now myself so I know exactly where you're coming from. And if our results match up, then I can no longer be a naysayer. And now I have to understand why what you did was successful and why my idea of this process, this protocol, this experiment, pathway is flawed. And this is what science is about, right? It's not about holding and maintaining the ideals and theories and hypotheses that we have, but it should be constantly changing, right? The laws of nature are still so elusive to us that we shouldn't pretend like we have it all figured out. Uh, in regards to the post-publication peer scrutiny, allowing your protocol, your simulation, to be associated with a journal, like I said, allows scientists to before they scrutinize, do it themselves. But also now, once they want to scrutinize, they can isolate or pinpoint an actual step of the process, a step of the protocol, right? Each of the steps has its own kind of hands that move that are recorded from the OP. And so now they can get down to a actual step-wise granular point and say, why did you use this concentration of this solution here? Why did you use this brand? What was the lot number? All of this is able to be kind of packaged inside of this platform, inside of the simulation. So it increases transparency so much that the peer review scrutiny is able to be much more pinpointed and allow it to be the experiment or the paper or the protocol to be dissected in a much more, I'd say, elegant and fine-tuned manner.
Yeah, it takes a lot of the questions, a lot of the blank spaces out of it.
Mhm.
Anybody who's dealt with reviews, oftentimes you get these, they're not inane questions, but they're questions that are not poignant to the actual scientific task, but more around it. And I think this kind of information transfer allows us to kind of skip those steps where people can see exactly what you did and then hone in on specificities that they have questions for in terms of how this came about and why you made that decision. Not so much, did you make this decision itself? So
Exactly.
efficiencies, yeah, efficiencies galore. Very, very excited about this kind of stuff. So it's clear that that there are so many questions, Sharon. I'm definitely gonna need to have you back for another episode. But I like to often talk about how DeSci leverages Web3, but is not fundamental, or Web3 is not a fundamental piece of DeSci. It can be switched out for other applications. Qure is using Web3 technology. Metaverse exists also beyond Web3, but you did mention blockchain security. And so to that end, what are you guys, is Qure running its own blockchain? Is it a DLT other, is another form of DLT? Are you using, leveraging an existing open source blockchain? Like what are some of the technical infrastructures that you have in place that you can share?
Not able to share too much on this right now, as it's evolving, right? We're really w- our our organization is a little different from most other DeSci projects in the way that our virtual reality product is powerful in the way that it is able to accelerate innovation and make scientists' lives much easier. Our use of blockchain really is for, as a beachhead for the smart contract functionality, right? Finding the right partners to partner with, those are the discussions that we're having today for our blockchain functionality. But the primary attention that we have and the primary attention that my advisors and co-founders keep bringing me back towards is let's focus on the VR, right? The blockchain, we have an immense opportunity to get into blockchain, to get into DLT, DeSci, and help many of the other DeSci projects, collaborate with them. But right now, it's a full focus on the VR product because without the VR product, our ability to impact DeSci and our ability to impact um, the future of science won't be there. So that's where much of our attention is right now is commercializing this. And that's where we're maybe a little bit different. Our commercialization, our model, our business model is almost a Web2 SaaS kind of model, right? But it is perfectly primed given our use of metaverse technology like XR, blockchain smart contracts, and AI to move towards blockchain and really provide a great amount of value and also offer an amazing ability to onboard scientists who are in industry, who are in academia, into DeSci with a product that provides value, first and foremost. So I think that the barrier of entry for traditional science to get into DeSci is a little high. So our approach to creating this DeSci platform is a bit of a slow burn. It's a slow roll until we have our virtual reality products completely set up, and that this truly becomes a platform, an ecosystem. And once we have an ecosystem and a platform, organizing that ecosystem, organizing that platform, promoting governance and uh, rules and regulations that are set by scientists, community standards set by themselves, that's where we want to go in regards to blockchain.
I think that's a really that's a really well measured approach. I would qualify what you're doing, what Cure is, as enterprise facing DeSci. And that often sits between the divide of Web 2 and Web 3. Takes a lot of the ethos, may not carry over the tech quite yet, but is, I think, essential for, as you said, bridging over traditional legacy science, but one that can, those that are at scale, that are operating at scale. We need that bridge. So I think this is a fundamental piece that is often overlooked. I think we sometimes get caught up in the narrative of, we're going to make a new system, but you need bridge builders to help bring people over to that new system and gradually. So I
Right.
completely agree with how you guys are doing it. I think it's a very smart and measured approach. So as we approach the top of the hour, I want to I have one more question, and I'm going to give you the open floor. And the last question is, what can we expect from Cure? So what can you share with regards to the roadmap in the next 5, 10 years?
Yeah, you so the next 5, 10 years. Wow. Okay. So, in the next 5 years, our goal is really to create a platform for scientists, an ecosystem, a home for science, right? Where you're not measured by the institution that you're at, you're not measured by the journals that you've published in, and you're not measured by who you know at funding agencies. You are measured by the the caliber and the reproducibility of your science, as should be, right? Some of the best scientists I know are not the best politicians. However, some of the best politicians become some of the best scientists, right? Uh, and that's the difference between talkers and doers. And there's room for both in science and in DeSci. However, in 5 years, that's our goal, is to create this ecosystem. And I try to relate to other founders and people in the DeSci community that why do we do science, right? Well, it's because we're curious, right? But we also know that our work and that the passion for our work leads into downstream to saving patient lives, right? That's why much of pharmaceutical research is happening. Medical device research. We want to help people, right, from as upstream as possible in preclinical research. So our goal is to connect all the world's preclinical researchers. And our next step in Cure is actually a step towards the clinical realm. This is a kind of a new announcement that our next kind of step is more focused on standardizing the um, manufacturing and tech transfer steps of scientists into scalable, reliable drugs and industries. Where now, if a DAO has created a novel therapeutic, a novel vaccine, anything amazing, a small group of dedicated people who want to change the world, they come together and they come up with a product that can really make an impact in people's lives. Should they have the option to sell it into pharma and industry? Yeah, they should. But should they also have the ability to have a decentralized ability to manufacture and create this drug that won't be priced 500% over what it should be, right? That doesn't have a 500% margin like these pharmaceutical companies do? Then that's up to them if they want to do that. And then we should give scientists the power to benefit the world like that. So our goal over the next 5 to 10 years is to close the gap between scientists and the clinical realm where patients are treated, by giving them the tools, resources, and decentralized atmosphere to make that change in the world.
And I would argue that I'm not argue. I would argue very strongly that that's essentially what DeSci is. It's similar to what open science is. It's giving more options to scientists to be able to leverage their capacities, their skills, their assets, their doings in the variety of ways that they so choose. And I think that's really what's been lacking is the that optionality. And I think we now have the playing ground to create these different options and these different modalities. Um, for scientists, for science curious individuals, but also for the patient populations that are the receiving end of this. And patients, as in those who are sick, but also patients of, I think we're all patients of science. We all are the endpoint users of science. And so being able to interact and have a say in how this is done and really understanding what is affecting us and who are the individuals that are doing these kind of things, I think it's very important. So as with every other person who we've had the opportunity to interview, my hats go off to you, Shar, for taking the initiative and developing this platform. I think your years of experience is something that is somewhat unique within this space. We need more individuals like yourselves who have had a lot of time in the traditional scientific space and can kind of meander through and understand that there is a need for a bridging aspect while still maintaining the ethos of the movement and the cause. So again, my hats go off to you. Final question,
Thank you.
and then I'll let you kind of wrap up. How can the wider community, the DeSci community, but also just individuals in general, help support Qure?
Yeah, you start off by following us on Twitter. I think that'd be a great place. But after you follow us on Twitter, be sure to reach out, send me a message, either on my Twitter or on Cure's Twitter. Go to our website, read our blog, and if you're interested in trying out Cure's Metaverse platform early on, be sure to reach out and we can start that dialogue.
Fantastic. So again, I'll just double click that. Follow them. In the jumbotron here at the top, we have some videos and links to their blog. I can tell you firsthand that Shar is a fantastic person to talk with. Very personable, very chill kind of person, and I'm looking forward to seeing you in person again, my friend, sometime soon, hopefully in Denver,
Same
if
here.
possible. And so with that, I will just kind of wrap up the space. So again, go help out where you can, go follow where you can. If you have a dream, you're a scientist or you're science curious, I think there's an opportunity for you to participate in some project in the DeSci space. If not, start your own. This is a very small space with a lot of really engaged individuals that want to see science changed for the better. So if you have an idea, you're more than welcome. Reach out to myself, reach out to Carolina, reach out to Shar. I see Brenna in the audience as well. These are all what I would call DeSci OGs, people who have been here from the beginning, if not the very beginning. Um, so yeah, I think this is a great place for us to grow our vision. So with that being said, everybody, I wish you a fantastic weekend. We will see you next week for our next episode. Shar, thank you again for coming on. This is a fantastic talk. And yeah, I wish you guys all the best. Have a great
Thank you.
weekend, guys.
Thank you all so
Absolute
much.
pleasure.